Great. Well, thanks everyone for making it here in person. I realize it's a bit of an unusual location. And thanks everybody as well online for joining. So yeah, it's my pleasure this week to introduce our speaker in the Maths and Life Sciences series. So Professor Bindi Brook has been professor in, I should have written it down, but your mathematical biology and in medicine.
Mathematical medicine and biology.
Yeah, yes, mathematical medicine and biology at the University of Nottingham. I've worked with Bindi for a few years now, mainly on all things respiratory, and Bindi's experience in that area goes much further than mine. She's worked at the micro scale up to some whole organ scale projects, which you're going to talk a bit about today as well, or maybe not whole organ, but
yeah,
more than the micro scale and everything in between. But yes, lots of nice mathematical biology that you're going to try and squeeze into sort of an hour. So won't take up any more time introducing. I'll just hand straight over to you. So
thanks very much, Carl. Thank you all for coming. I think it's been a few attempts at getting me here. I'm finally here, so it's nice to be here. First, just wanted to start with acknowledgements, my funding, but mainly the people that have done the hard work here. My students, Ewan and Alice and Melanie, some older stuff from Hannah, Jonathan and Michael. And also to say that a lot of some of this work sort of started way back when Igor and Oliver were in Nottingham. So I'll give a little bit of history as well. And then people who do the experimental work at the Centre for Respiratory Research at the University of Nottingham. And finally, just a quick plug for our network, BioReam, which is about all things respiratory and modelling. We don't have much time left on it, and I think maybe quite a few of you already know about it, but if not, then look it up, and it'd be lovely to see you at some future event. Okay. Let me start by telling you a little bit about asthma. For those of you who don't know that much about it, it's a chronic lung disease. It affects about 7 million people in the UK alone. And severe asthmatics suffer exacerbations, frequent hospitalizations, and so it's a significant burden on the NHS. There's currently no real cure, and by that I mean no complete remission of the disease. We only really treat symptoms, and this is partly because of multiple phenotypes of the same disease. So the disease itself is characterized by these three things: inflammation, hyper-responsiveness, and remodeling. When we breathe in an allergen, there's an immune response. White blood cells come into the airway to sort of try and deal with it, and then they tend to hang around in asthma longer than they should, kind of causing inflammation of the airways. Hyperresponsiveness refers to the rapid contraction of the airway to much lower doses of a contractile agonist that gets released as a consequence of that inflammation. And then over longer periods of time, we actually get what's called airway remodeling, which is structural changes to the smooth muscle and to the and extracellular matrix and a bunch of other stuff. So I'll just show you here if my cursor works. I'll just point. So what you see here on the left-hand side is a normal airway, and the pink bundles are the smooth muscle, and extracellular matrix is a little bit harder to pick out, but you can see that in a severely asthmatic cross-section through an airway, you get much thicker smooth muscle bundles, a lot of epithelial cells, and then an irreversible narrowing of the airway. This picture here, or this movie, if it's working, I think it was working, is of what's called a precision cut lung slice, and that shows the acute changes, sort of what might happen during what happens when you apply a contractile agonist at increasing doses, and you can see the increasing fraction of the airway. This is from a mouse lung. Okay, so let me just describe some short timescale effects first, before going on to talk about the longer timescale effects, the remodeling, which is the main thing of this talk. essentially, when we breathe in a contractile agonist, as I said, the consequence of that is to cause interactions between protein filaments inside the smooth muscle, which then causes the smooth muscle to shorten, the smooth muscle cell to shorten, and the airway to narrow, causing sort of the effects that we observe at the organ scale in terms of wheezing and coughing. At the same time, of course, we are constantly breathing. So what's happening there is you're applying strains to the airways, which are then feeding down to the muscle cells, perturbing that interaction between proteins, perturbing those things, and thereby having a sort of an effect on the molecular scale. So this is constant interplay between what's going on with the molecular interactions and also our organ scale breathing. Okay, so one other thing I should add is that there's increasing evidence is that growth factors can get activated as a result of interactions between smooth muscle cells and extracellular matrix. Normally it's stored in latent form, but increasing constriction is thought to now have a potential impact on longer term airway remodeling. Okay, so some time ago, we developed a mechanical model of compliant airway coupled to a cell model, which had these protein interactions going on. And we have also incorporated within this airway stiffening of the extracellular matrix, the collagens that are inside the airway wall. So what I've got here is a result of such a tissue mechanics model that incorporates those two things. And what you see here along here is what happens when you apply an inflating transmural pressure, so internal pressure minus external pressure, and what happens to the lumen radius as you do that. And we've calibrated that model to experimental data. So that's what you're seeing here. This is in the absence of any contractile agonist. And then because we've coupled it to the tissue mechanics model to a cell model, we can now investigate the response of that airway to increasing levels of contractile agonist. So what you see here now is that for the same inflating transmural pressure, you can't open the airway as much as you used to be able to do. Okay. So what's actually happening inside? This is a finite thickness airway. I'll get to the details in a minute. You can actually look at the circumferential stresses and the radial stresses within the airway. And here I'm plotting the circumferential stress as a function of the radius through the airway. And you can see here how we get these quite non-uniform stress distributions as you first, when you inflate. So this is in the inflated state. First, when you just apply inflation, and then a response to that as you increase the contractile agonist. And you can see, and eventually, if you keep increasing the contractile agonist, you actually get into the compressive forces near the lumen. The compressive stresses near the lumen. So here's an example then of a contracted one, circumferential stresses shown as a heat map, and then what happens when you increase when you then inflate that contracted airway. And again, you're seeing this really dramatic difference in the stress distribution. Okay, so what we thought that we would like to do is, go back slightly, is to think about how this microenvironment of these stress microenvironments actually affect airway remodeling or structural changes in the airway over time. So this is now kind of really the meat of the rest of my talk. What we do is we imagine that within the airway wall, we have smooth muscle cells that exist in either a contractile phenotype or a synthetic phenotype, and that these can switch between the two. and only the contractile phenotype can die naturally, and only the synthetic ones can can proliferate. And then the synthetic phenotype can also synthesize extracellular matrix. So this is this part really is stuff done what I did with Igor many years ago, and then we've developed it further with later on. And this is what I described later. Oh, I should say that in Igor's work, we did look at how inflammation affected switching. And so we include that here too. And we also look at how inflammation affects extracellular matrix. These are all processes that we know happen biologically, but from sort of separate experiments that we're bringing together here. Now, inflammation causes contractile agonists to be released. That then generates an active tone, i.e. the sort of circumferential stresses that I talked about. And that, in turn, through mechanosensing type or mechanotransduction type processes, those stresses can affect things like proliferation, switching, and apoptosis. And so here, wherever you see tau in the things that I'm going to talk about, this is the sort of coupling between the sort of mechanics of the airway model and the sort of biological processes. And then, of course, the changes that result in these constituents feed back to the tissue stresses, of course, because the more smooth muscle you have, the composition of your airway becomes different. And then finally, one can apply, so from an experimental point of view, we can apply inflammatory agents or contractile agents to an experimental model. In everyday life, we're typically subject to inflammatory agents through things like toxins and so on. Okay. So our model then of the airway, based on that sort of picture, essentially comprises 4 constituents. This contractile smooth muscle phenotype, the proliferative phenotype, an extracellular matrix, and then some interstitial fluid that just remains constant. We assume the airway is axisymmetric and is compliant, and we use a hyper-elastic formulation, and that there are fibers embedded, circumferential fibers embedded in the airway wall to represent both smooth muscle and the collagens. In reality, what you have is the smooth muscle wraps around the airway in a helical fashion. So if you were to cut a cross section through it, you would actually see bundles like this, a bit like those pictures we saw on the histology. But because we're going to make the life simple for ourselves, we're just going to assume that everything is axisymmetric and that these fibers are not helical, but just circumferential. Okay, so the other thing I should say is that we're going to model the growth of the airway and the elastic deformation of the airway, and we're going to use what's called a standard thing called a multiplicative decomposition, where we have a growth deformation and an elastic deformation. But we're so we start off with some initial unremodeled airway, and which I had in my previous picture, so we've got some kind of reference radius or undeformed configuration, and then we have a grown configuration where those biological processes cause such as proliferation of cells, synthesis of extracellular matrix is ongoing, and this adds volume to the airway wall. We assume that the outer radius remains fixed, and all of the growth kind of contributes to inward growth. These are all just assumptions to make our model easier to solve and be practical. So from now, so this is what's called a growth deformation, and so wherever you see radius psi, it's in this configuration that I'm referring to. Okay, so we have mass balance for essentially a multi-phase model because we've got these four constituents, and we assume that all of the four constituents have the same velocity, same growth velocity. We assume that there's no voids, which mean all the constituents add up to 1. And obviously the constituents that I'm talking about, phi, are functions of both psi, the grown radius, and time. For each of the four constituents, we can add up, well, 3, because they're the only ones that are changing. The water remains constant. Add up the 3 constituent equations for the 3 constituents, you end up with this equation here. S represents the source terms that represent the biology that I described. So we now have an equation for tissue velocity. Okay. So the source terms are functions of our 3 constituents and also inflammation and stress. Just something to bear in mind. And then we end up with a moving boundary problem because we've got both of the inner wall, which we can write down as d psi n dt equals v at psi n. And then the inflammatory factors we can model using a simple time-dependent term, which is how we challenge our model, and then some natural resolution. And the contractile agonist, K, feeds into the mechanics model, which I'll show you in a second, but we can apply that exogenously. It resolves through binding with receptors and so on, and it can be produced either through inflammatory factors or through some stress-dependent term, this activation of growth factors, which act as agonists as well. Okay. So how do we work out tau? That comes in through what we call our elastic deformation. So now whenever I talk about radius, I'm talking about a deformed radius, R. So the types of forces that are at play here are the transmural pressure that I've talked about, but also a contractile force generated by the contractile agonist. So the equations that are relevant here are we assume the tissue itself is incompressible, that we're in a plane strain, so that the airway is held fixed at the 2 ends, so there's no axial deformation, and then we want to solve balance of linear momentum, which in the axisymmetric case just boils down to this equation. And then we've got a bunch of constitutive laws for each constituent. So for example, for the collagens, we assume that there's a sort of a stiffening response, and the constitutive law sort of describes that. We have an underlying near-Hookean matrix for what would be elastin, so to speak. And then for the smooth muscle, again, these all act around the fiber, around fiber directions, and nothing. Oh, and within that constituent, we have a way of modeling the effect of the contractile agonist. Okay, so just if you want any details of all of that, it is in the paper I cited. But for now, I'll move on. If anybody has any questions, I'm happy to answer them. But essentially, what I wanted to talk about for a few minutes was how, using this model, we've been exploring the parameter space. It's a huge parameter space, but we sort of focused on a few key things. So, for example, here we've essentially looked at what happens when we challenge the airway with just contractile agonists. So this is, for example, inhalation of allergens, which then cause contractile agonist to be released. And here we're just modifying the agonist magnitude, so how much agonist is being inhaled and how quickly it clears. So those are the two parameters. That's the parameter space we're looking at. And what I'm showing you here are surfaces starting from some, so the blues are some initial starting radius, and what this shows is how much that inner radius has changed for different values of these parameters. And this picture here shows how quickly the contractile agonist resolves in time. Okay, so we can so picking just one point on that surface, we can look more carefully at what's happening to the actual airway. So along here, I'm plotting time, and the thickness of these things reflect the radius of the, the thickness of the airway itself. And we can look at different things. So for example, here on the top, I'm plotting the contractile agonist. Here I'm plotting the proliferative phenotype. And here I'm plotting the circumferential stress, and you can see how that varies across the airway wall. So a few things, excuse me, a few things to note is that, so here we start challenging. You can see the growth of the airway wall. You can see where most of the proliferative cells are located. And then importantly, we see that even after challenges have stopped, we still continue to get some remodeling ongoing. Okay, so somehow we got into some kind of dynamic, some dynamics that continue this process. And then what we can then do is we can sort of also zoom out, so to speak, and look at the whole mechanical behavior, the mechanical behavior of the whole airway. So remember those pressure radius curves I showed you right at the beginning? That's what I'm plotting here. So the faintest curve is the curve right at the start of the remodeling, and how the changes in the constituents affect those pressure radius relationships, or that the sort of what some people in the field think of as a tube law, how that gets modified during remodeling, and then how once you stop challenging the sort of what the mechanical property of the remodeled airway has become significantly different from what we started with. So one thing you might ask is, it's not easy to get these types of relationships on human airways, because you can't take out a whole airway and test it and keep them alive. But this is kind of the equivalent of what might be a pressure volume curve. And that's something that we measure all the time in patients. And so one thing I'm very interested in is understanding how this modified mechanical property in the branching network affect pressure volume relationships in people who have severely remodeled airways. And this is work that I'm sort of starting with Carl, and so I don't have anything to show you in this presentation here yet, but this is, yeah, this is something we're looking at. Okay, gosh, how have I been going half an hour? All right, yeah, and so details are in this paper, and I just then want to finish by talking about how, by exploring some of these parameter spaces, we've sort of identified certain asthma phenotypes. And what I mean by that is different manifestations of what people think of as asthma, but to have kind of slightly different underlying fundamental biology. So for example, in severe eosinophilic asthma, we essentially have resistant eosinophils, type of white blood cell. There's some resistance to corticosteroid. There's extensive remodeling and basement membrane thickening, and that the basement membrane is a thick extracellular matrix, bit of extracellular matrix between the epithelial cells and the rest of the airway. So here we've actually got a 2-layer model. I'd previously switched it off, but I've kind of switched it on in this example. And what we see here is that by applying frequent inflammations, inflammatory episodes, and impairing the clearance, which is what this parameter space here shows, and by having an elevated agonist release as a result of inflammation, we end up getting these predictions. So there's a thickened airway wall after challenges. Remodeling continues after challenges. Mechanical properties are significantly altered, and we have this thickened basement membrane, which sort of are associated with some of these clinical features. Similarly, there's another type of phenotype called granulocytic asthma, which I don't know if I'm pronouncing it correctly, but essentially what we're doing in this particular case is we're keeping the inflammation minimal, but we're enhancing the feedback, the mechanotransductive feedback between smooth muscle, between the stress and activation of agonists, and also making the smooth muscle more hyper-responsive. And what we see here is that we, if we look at the, so along here is this hyper-responsiveness type parameter. If we look at some point on that parameter space, we get something that looks like this. Here I'm plotting the contractile agonist. But if you just move the hyper-responsiveness slightly along that axis, then we end up with this runaway feedback loop where we've got this really rapid increase in airway wall thickness, and the agonist takes a long time to resolve, suggesting that there's this persistent contractile tone in the airway. So I think I sort of said all of that with severely altered mechanical properties that you see up there. And this sort of fits with the clinical features of this particular phenotype, where you actually have just normal eosinophil counts, and so inflammation is minimal, and yet you have this persistent tone, which could be somewhere in this, because of this level of agonist that is being released all the time, even post-challenge, and such people with this type of asthma can get very severe bronchospasm and need to be hospitalized as a result. So this is just a really exploratory type thing that we looked at, and we would need real data to really sort of identify where we are with that, and I'll come to the data part in a second if I have time. But what I wanted to get onto was this idea of homeostasis. So obviously there's this sort of runaway feedback loop that we sort of identified here, but whether that's but that is unlikely to be something that happens in reality. There are probably regulatory mechanisms that are going on that limit that, and we may not have incorporated enough of that within our model. And what we really wanted to do was try to identify potentially stress-based mechanisms that could limit that. So we started to ask, given the very different types of circumferential stress distributions one can get, depending on the particular phenotype that we're looking at, or the particular set of parameters that we're looking at, is there some kind of stress-based regulatory mechanism that actually maintains homeostasis in healthy controls? The other thing I was also going to say was that all of these simulations that I've shown so far are somewhat arbitrary initial conditions, in the sense that they satisfy the homogeneous steady state. So if I go all the way back to my ODEs and set my time-dependent terms to 0 and my spatial terms to 0, then we started our models from a homogeneous state that satisfied those equations, and then looked at things go from there. But that's somewhat arbitrary in the sense that they don't satisfy a dynamic steady state where the tissue does not grow. That only satisfies the multiphase model, not a velocity equals 0 at the inner boundary, i.e. you would get thickening if you challenge regardless of anything else. Well, you could get thickening even if you don't challenge, depending on the parameters that you're at, if the steady state is unstable. So what we really wanted to do was get to a steady state, identify steady states that were homeostatic in the sense that you also don't get thickening, the velocity of the inner wall remains 0. So to do that, we said, let's forget about inflammation and forget about exogenous contractile agonist. We have some kind of closed system where everything is in some kind of steady state, or we'd like to find some kind of steady state where you don't actually have any thickening of the airway wall. So what we mean by that is we want V at the outer radius to be 0, but we also want the velocity, the tissue velocity at the inner radius to be 0 as well. So having removed the inflammation and agonist, exogenous agonist, we just make life a little bit simpler for ourselves and say that the stress-based mechanism only comes into play for proliferation and apoptosis. And so essentially what we do is we say, let's imagine our proliferation rates depend on circumferential stress in this way. Your stress gets too much, the cells want to proliferate to relieve that stress. The stress gets too small or compressive, the cells die in order to relieve that stress. And that there's some preferred stress window in which the cells sort of like to be. So that's all we're doing that's different to previously. So all our equations are identical, except for we have to add a little bit of diffusion because it doesn't mean anything physically as far as we can see, but it just makes our numerics a bit easier to deal with. So everything is exactly the same. Some terms disappear because we don't have any information. And our source terms now have just the stresses in the apoptosis and proliferation terms. Okay, and now our boundary conditions, these are the same as before, but now because we've got a second order derivative, we can also apply two zero gradients Neumann conditions at the two boundaries, whereas before we could only impose one. Okay. So we started out from some initial condition of some fixed airway radius, with some constituents distributed in the airway in this sort of manner. And then we just ran the model and looked at what happened for different values, for some fixed value of delta, but different values of tau naught. And so these are the things to remember. Tau naught is the mean stress, and delta represents the window, the preferred stress window. So for some fixed value of delta and increasing tau naught, we see this sort of behavior. So what we see here is that we're actually approaching, or we get to a steady state for those orange curves, but not for the blue curves. And indeed, if you keep running those blue curves longer, we don't reach a steady state. And we can plot those. So we can now explore the parameter space tau 0 delta, and we find that there is indeed a region above some boundary and a region in which we get steady states, but a certain region in that space where the airways continue to keep remodeling or narrowing. So what's going on inside those airways? So one thing to to point out is the circumferential stresses. So at those final times, I've plotted the circumferential stress across the airway for the same value of delta and increasing tau naught. And what we see is that for the blue curves, so for the thickest blue curve, I've plotted the stress window. We see that the stress does not span the entire window. However, for the orange curves for which we do have steady state, and certainly the thickest curve that I show at the top here, the stress curve spans the entire window, or rather spans the stress window and has some regions below and above it. The corresponding contractile smooth muscle distribution varies in this way, and I'll get to that in a second. And the velocities now, the tissue velocities, you can see how they're responding for the steady state compared to, so that's the orange curves, compared to the ones where you can discontinue remodeling. So you see that these velocities here at the inner radius are significantly negative, which means that they continue to remodel. But these ones have settled such that the velocities at the inner radius are 0. So what's going on? Well, if I take the last curve and look at the circumferential stress initially when we first started out, the evolution of that airway causes the constituents to change in such a way that you end up with this thick blue curve here. On the other hand, if I start with the same initial condition, but up at the top, the thickest orange curve there, and look at the evolution of the stresses for increasing time, you see that you get this sort of thing happening. So what we think is going on here is that there's, by choosing a particular set point, where the constituents are evolving such that, how best to explain it? As they're evolving, the stresses are being modified, but in these examples, they never, the stresses that are above here have the increased proliferation. Stresses below here have the increased apoptosis. And because there's nothing in this region for these curves, there isn't a balance between the proliferation that's going on at the outer edge of the airway compared to the apoptosis that's going on elsewhere. So you never get a balance in these cases, but you do here because there's enough of the airway with high enough apoptosis and high enough proliferation that you get balance between those two processes. Sorry, that's just me trying to say that. And if I now take one of those steady states and just look at dependence on the initial conditions, so here I've just taken one of the cases where it definitely goes to some steady state and then just varied the initial radius, then again, we see that they all go to the same steady state. And again, by looking at the circumferential stress, we can see that because they start out with some region below and above this preferred region, they all settle to some circumferential stress that again has this balance between proliferation and apoptosis. And then for the unsteady states, the ones that were blue curves before for different initial radius continue to remodel as in the previous example. So how do we understand that beyond me waving hands and saying there's regions here and here that balance? If we go back to our velocity equation and integrate that, in principle, we can write down what that velocity looks like in terms of this function Q, which is the source, which is the sum of the sources of the different constituents. then we can see that from this expression, we can get a velocity at the inner radius to be 0 if either psi in is equal to psi out, which is when the airway is completely thinned to nothing, pretty much, or if the integral between these things of q times psi is 0. So what does that mean? It essentially means that if you were to plot q at any particular time point, which is the sum of the sources, which depend on the phase, which depend on space, your curve might look something like that. And the area, so where the blue and red areas are equal, you would then end up satisfying this equation here. And sure enough, we can plot, for instance, the integral at final time points for a couple of examples for increasing tau 0. And we indeed see this sudden drop that matches that map that we showed originally for velocity in the tau 0 delta space. So I should just say, so this is a steady state, clearly, but it looks like it's an unstable one because all of these pictures here, as we get closer and closer to the outer radius, they still always come back to this steady state. So we think that if Rn equals psi equals psi out is a steady state, it must be an unstable one. But we haven't done any of the stability analysis. It's quite, so far it looks like it might be complicated. How much time do I have left? Okay. Okay, so just to finish this part, let's take an actual example of a steady state. We have a velocity profile across the airway wall, looks like this at steady state. And for that particular steady state, our proliferation and apoptosis rates, proliferation and apoptosis rates look like this. So you can see, of course, that here there's going to be effectively more death, and here effectively more proliferation potentially. And what we have, of course, is that all these velocities are positive. That means that the tissue velocity, so the cells actually and tissues are moving in this direction, but this increased proliferation keeps the airway sort of supplied at this end, and the increased apoptosis keeps them dying at the other end, so to speak. And sure enough, the profile of the contractile phenotype, for example, I could have plotted other ones, looks something like this. And if I take that net growth term Q and plot it, then sure enough, again, you can see that it perhaps it matches with the things I showed you previously about the integral under this curve. And in getting to this steady state, we've effectively got the stresses to behave, to evolve, or rather the constituents to evolve in such a way that it actually settles to a steady state that gives rise to this, what we think of as a dynamic steady state or homeostatic steady state. So what we'd quite like to do now is perturb these and see by challenging with inflammation and see kind of how they respond, whether they're stable or unstable to these perturbations. Maybe they need to be large enough. So that's kind of where we've got to. Does anybody have any questions at this point before I move on? Yeah.
Not earlier, but you talked about two distinct types of asthmatic airways.
Yes.
Are there things that can be measured that would, connect to the model, such as if you were to excise an airway and look at patterns of residual stress differently. Would there be measurable differences there, as you were predicting different stress profiles?
Yeah, it's a good question. So in those simulations where I predicted these different types of asthmatic airways, I don't know if we ever got to any kind of steady state, but equally there's these potential residual stresses that might be present. Oh, the contractile tone, for example, that potentially is present all the time. So I think I've asked this question of quite a lot of physiologists, and as far as I've found out, nobody's done that kind of test where you cut the airway like they have done for cardiovascular stuff. I really want to get people to do that, but not yet, because many of the exercise airway studies are not done on human airways. They're done on animals that don't spontaneously have asthma. And in mice, I'll show you a little picture in a second, that the airways are so small, I don't even know that they could do that. But I'll tell you a little bit about some of the data that we've collected, and hopefully we're going to connect to this model and sort of look at whether homeostatic states in control mice versus challenged mice might look how they might look. So so a very brief and quick look at some of the data that we've collected. So this was an experimental model that we ran alongside our model development. So mice are challenged with something that we know causes airway remodeling, and then after a certain number of challenges, we stop and we examine the airways at different time points post-challenge, and some of them have had nothing done to them, and others have had the challenges. So we've got a set of control and over mice. Over albumin is a protein that causes remodeling, and we are able to essentially show, we have shown from the data collected there that over time, the smooth muscle increased rapidly between start and maximum remodeling time. So that's after the challenge is finished, and then seem to resolve almost to baseline, whereas the extracellular matrix started going up and remained elevated even post-resolution, and the epithelial cells went up and remained elevated post-resolution. And then additionally, we were able to look at spatial distributions of both smooth muscle and extracellular matrix as a function through the airway wall. And then because of the numbers of airways we managed to collect, we were also able to stratify according to airway size. I want to show you all of the data, but essentially the pictures look something like this. We have to do some mixture of semi-automatic and user expert manual sort of stuff. We got a whole load of ground truth. We managed to get about 1,000 airways in each of these two categories, for each of these two categories. So this top here shows collagen, is a collagen stain. This one here is a smooth muscle stain. And now we're looking at using deep learning to classify those more easily and also to segment them more easily. This is work from Melanie Young, a current student. And just very quickly to say that we now have spatial distributions across the airway wall that would then allow us to do some of the connect it to our model, and lots of data also separated by mice as well. And then other things we have also got data for, and that is the inflammatory response, because every single mouse had some of their fluids tested for various types of white blood cells. We've also got these precision cut lung slices telling us how the what the contractile response looks like, and we've got these mechanical properties as well for mouse airways, and those were really tricky to do by my postdoc. That links the mechanical properties to the amount of smooth muscle, collagen, and elastin that's in there. So yeah, that's just some of that data, but I don't want to spend too much time on that. I don't know if I've got 5 minutes left, maybe. Okay, so for now, I just want to finish with some conclusions up for this part, which is just to say that our models seem to suggest that there could be some self-sustaining remodeling under certain conditions, that the mechanical properties after remodeling potentially mimic hyper-responsiveness, and that's something that I failed to highlight. If you recall, our initial pressure radius curves post-agonist looked like they'd been shifted that way, and the same sort of thing is happening in remodeling. So there's some potential link there between hyper-responsiveness and remodeling, or how it's measured or perceived. But phenotype heterogeneity could arise from mechanical coupling, as I've shown, might help us identify appropriate targets in time. And I just want to say that some of the things that we've been looking at so far have all the things we've shown you so far have all been about thickening of the airway. But we can imagine, or you can imagine, that if I chose some slightly different parameters, that I could end up in a state where I had airway thinning rather than airway thickening, as in my psi in equals psi out might have been a stable state instead of an unstable one. This is just a hypothesis. So we're asking the question, could it be that we could maybe demonstrate airway loss using a similar type of model that happens in COPD? So in COPD, you get small airway loss. Is that through thinning along similar types of ideas? And we really need to link to our experimental data. So that's really the next big step. Finally, I just want to show a few very quick movies on some emphysema modeling we've been doing along similar sort of lines, where we've got, but this time we're not doing it in airways, we're doing it in individual springs of a spring network. We assume that there are processes going on in each spring. The spring network represents, if you look at a cross-section through lung parenchyma, the tissue of the airways, of the tissue of the lung, then it looks something like this. And so we just wanted to, there's lots going on here, but we wanted to ask the question, given some injury that happens as a result of smoke or in another disease called LAM, lymphangioliomyomatosis, you get injury as well in a very not in a similar way, not through environmental exposure, but through mutation, certain mutations. And essentially you get a collagen degradation and elastin degradation, causing those alveolar walls to weaken and rupture over time. So we've essentially, for each spring, we have a little model that sort of looks at the response to that injury. But ultimately, it's the release of proteases that we're interested in. Don't worry too much about the other details. And here's just some simple models we've run with that simple ODE system. And what we're showing here is the release of proteases for some I think there's about 7 or 8 randomly injured springs in here. This is work done by Alice Perrault at the University of Nottingham. So you can now see that some of those injured things are now breaking as a result of exceeding some strain threshold, and the consequent changes in stiffnesses and stresses, which you can't see very well, but you might pick out a few things there. Okay, so that's when I start off with all homogeneous natural lengths and stiffnesses. If I run if we run the same thing, or Alice runs the same thing with heterogeneous lengths instead, you find that you get quite a different pattern of damage. Get there eventually, and I promise I'll stop after the next slide. And that's shortly. But you see a slightly different, so you start to see more damage elsewhere, whereas before all of the damage was occurring around the initial big injury. And you see kind of a redistribution of some of the stresses. Oh, sorry, one more. Maybe I'll stop there and just show you that we can look at all the individual springs and look at those proteases and strains and stiffness and stresses. And we see that a lot of the initial breakages are happening obviously for the initially injured springs, but then other neighboring springs, so the green ones, are getting damaged. And And so these fast timescale things are a result of the tidal breathing. And yeah, a lot more to do here to quantify all these things, but I thought it'd be fun to show that because I know that Oliver's and with Carl are doing some stuff on this. Okay, work in progress. I don't have a lot of real conclusions. These are just some observations. Thank you.